Thank you, and welcome everyone to Foresight's Health Extension group sponsored by 100 Plus Capital. And today I'm super excited to have Irina Conboy here, who's going to speak about tissue rejuvenation via plasma dilution. And Irina was a while back co-chairing an health extension technical competition that we had. And so I'm really excited that you were able to join us again. I know that many people here are very excited for what you have to tell us. I'm going to kind of like backtrack all of the questions for later and just have you start so that people don't get too overexcited. And then I'm going to collect questions here in the chat as they're moving in. And I'm going to post your biography and everything about this group in the chat so that I don't take any more time away from your presentation. Thank you again from all of us here. And I think you can go.
Yeah, thank you guys for inviting me. So let me share my screen. Yeah, hope you can see it. And so my talk will be about plasticity of age and how we can prevent, attenuate, and possibly even reverse age-imposed diseases as a class, not one by one, which was not successful, but all together. So proverbially speaking, an antibiotic against aging. What you can see here is exactly what I see from my office at Berkeley. It's pretty nice courtyard, and unfortunately, I was not able to go there because of the pandemic for quite some time. So this slide is introduction into how we understand, or rather do not understand, the causes of aging. And we know that with chronological age increase, there is dramatic increase in organ deterioration and disease, which at some point becomes catastrophic. But very interestingly, this chronological age is very different for mammals that are similar to each other in many parameters that we consider to affect aging. For example, rats live only 3 years and squirrels live 20 or 30. They are very long-lived mammals. And even though the rats and squirrels have similar size and metabolic activity, in fact, if anything, squirrels have higher metabolic activity, higher metabolism than rats. They are mammals and they have very similar diet, but yet squirrels live 10 times longer. And so by the time the squirrel is 3 years old, it will be a very healthy animal, but the rat at 3 years old will be very sick and really aged animal. And so to us, first of all, it tells that we do not really understand what controls the rate of mammalian species aging. We don't, even though we have some theories. And secondly, that it is not simply the entropy or the rate of damage that controls it, because rats and squirrels really live in the same entropic environment. So perhaps it is the capacity for repair. So damage is something that we cannot change, but repair or efficiency of repair we can change. So here is another cartoon viewpoint on what I mentioned. So if you look at the young person and an old person, the degree of damage will be the same. In fact, an old guy who takes care of himself or herself perhaps has less damage if they have healthy diet and wear sunscreen, as compared to young people who may be basking in the sun and go clubbing every night and so forth. However, the rate of repair is significantly higher at the young age. And so perhaps if we can improve repair in an older individual, we can then rejuvenate them. So this is kind of the general conceptual introduction to my talk, explaining what are we doing and why. We believe that we cannot really chase down the entropy itself and reverse it at every single minute detail. Entropy will persist, but we can attenuate it and offset it by improving tissue repair and regeneration, and that is the key to longevity. So I will speak a little bit about our experimental systems. We look at mesodermal rejuvenation, endodermal rejuvenation, and ectodermal rejuvenation. And those are the three germ layers from which our bodies are built. So we really start with mesoderm, ectoderm, and endoderm, and then those differentiate into various organs and tissues. And so it is important, if we look at multiple tissue rejuvenation as a class, to make sure that all three germ layers become rejuvenated. For muscle, for mesoderm, we look at muscle, and we look at fibrosis, inflammation, poor regenerative responses. And we are able to show that old muscle, for all practical purposes, is turned into young muscle, where fibrosis and inflammation are decreased, and tissue repair after injury is now effective, moreover, by the old tissue stem cells called satellite cells. And then for endoderm, we assay typically liver, where we also look at fibrosis, adiposity, and poor regeneration. And our experimental systems rejuvenate old liver, turning it into young-like, with fibrosis and adiposity reduced, and hepatogenesis being increased. And then for ectoderm, we look at the brain, which is shared by many of our colleagues. Everybody is interested in rejuvenation of the brain and cognition. Old brain is characterized by poor neurogenesis or formation of new neurons, increased neuroinflammation, and also that leads to lack of cognitive capacity and memory. And we show that our approaches now rejuvenate the old brain, making it significantly younger, such as that neurogenesis is improved, neuroinflammation is diminished, and cognitive capacity is enhanced. And I put 4, 5, 6, etc., because we pretty much now analyze multiple hallmarks of aging and rejuvenation. So the axiom is that when several key organs become significantly younger, then the entire animal will be also younger and healthier, and it will live longer. And I bring this up because many people are asking me, did you do lifespan studies or health span studies? And even though I will potentially answer it, and we are on that path, you should realize that we are just the organ system composition. So if your muscle, liver, brain, bone, immune system all become younger and healthier, by default, it means that you yourself are younger and healthier. And if you are younger and healthier, or the health decline is now diminished and health span is increased, you will live longer. There is no other way around it. And then the opposite is not actually always true. So here is this image of a very old sick mouse, and even it is very old and sick, it is still alive, so it will be in a positive column if people are just looking at the lifespan extension. So many lifespan extended animals are quite miserable. So actually I have to apologize because I opened accidentally a wrong talk, which is slightly outdated. So with your permission, Alison, can I exit from this sharing and open the more? Yeah, thank you. Yeah. Stop share.
In the meantime, I'll let some more people from the waiting room in. And since, can you tell us what was the outdated talk? When was that? Like,
that was just a couple of months ago, and now we actually have additional publication that I wanted to demonstrate here. So just please bear with me.
I'm going to share in the meanwhile a few ways in which you can plug into the bio.
Yes. Okay, just to make sure that I have it all here. Yes, this is it. So, sorry. So it's just 2021 talk instead of 2020. Yeah, so let me return to Zoom. And we will go seamlessly from the previous slide. All right. Sorry guys about that. So it is because, we are all overloaded with Zooming and with so many remote talks. And I'm happy to report that we are publishing every couple of months on these discoveries. And so I just wanted to give you the most updated version. So we showed quite a while ago that aging is malleable for liver, muscle, brain in the first study that we published back in 2005, and also for neuroplasticity, cognition, spinal cord rejuvenation, kidney, bone cartilage, etc. And those additional pieces of work were continuation of our initial research on heterochronic parabiosis by our colleagues. So this is the final slide, which is a Halloween party at Bach Research Institute in Novato. And the slide is by Eric Verden, who is the director of that research institute, which illustrates heterochronic parabiosis, where an old mouse is physically sutured to a young mouse. So they share blood and they also share multiple organ systems. And while many people interpreted this as just blood sharing, you should realize that now this old mouse has access to young heart and lungs, and therefore just the oxygenation of organs will be better. And the old mouse now has better GI tract, so it will absorb nutrients better. It will go to food and water more frequently and exercise because it has to run around with the young guy. So there are numerous changes which are unrelated to the secret sauce in blood. And to make the system better controlled, we developed this small animal blood exchange procedure and apparatus where we exchange only blood, so the mice are not physically connected to each other. And this procedure takes about half an hour, and then they recover, and we could see what happens to their organs and tissues, are they younger or older. Moreover, of course, this procedure is not FDA approved, cannot be translated to clinic, but heterochronic apheresis is FDA approved, and it's called plasmapheresis for people. It was not previously used for aging and rejuvenation, but rather for blood diseases and autoimmune disorders. So once we published on what happens with heterochronic parabiosis and more recently with heterochronic blood exchange, typically our publications are highlighted in The Economist and other popular press, and everybody interpreted all this in the same way, that young blood is a medicine, and here is an army of old people who tried to steal this young blood. But that is actually not the case. And so to give you an example of why that is not the case, I show you one outcome of heterochronic parabiosis where young and old animals are physically sutured with each other, and blood exchange when we only exchange blood. So what you see here in green dots and red dots are the newly forming neurons or neurogenesis in a particular part of the brain in the hippocampus, which is called denta gyrus, and it looks like this V region with many blue cells. So at the edge of the V, there are neural stem cells which continuously make neurons even when mammals are already adults. And there is dramatic decline in this neurogenesis in the old animals. There is only maybe less than 25 neurons are made, as compared to young animals, which make more than 1,000 of them in this specific assay. And then after parabiosis, now old animals made 50 neurons instead of 25, and young animals have a diminished capacity to make neurons when they are sutured with old mice. So we discovered this back in 2004 and shared our unpublished data with Tony Weiss-Correa laboratory, and then they pretty much republished the same result 6 years later in 2011. So initially we wanted to include the brain studies into our paper in 2005 Nature on muscle and liver and brain, but we could not include them because we had too many suggestions to do additional experiments, and I already have my new job at Berkeley. But we are happy that Tony's lab picked up on those discoveries and confirmed and republished them. But now if you what happens when you exchange only blood and animals are not sutured, you have the same dots here and the same V region, can see them very clearly. But now, even though the young animals have better neurogenesis than old blood dramatically inhibits neurogenesis, dramatically. But young blood, old exchange with young, does not rejuvenate it. So if you suture young mice with old mice and they have environmental enrichment and more exercise, their brain becomes younger. But if you exchange only blood, it does not become younger, which is quite interesting. And then if you look at their cognition or short-term memory in this interesting test, which is called 4-limb hanging test. So mice are suspended upside down from the mesh wire, and they have to have coordination and grab to the wire. And then they don't want to drop down, so they have to remember that this is not good for them to just let go. They have to keep grabbing to the wire. So in this test, which is similar for us to learn how to walk on a tightrope, for example, if we are learning how to walk on a tightrope. In this test, initially, there is not much difference between young mice and old mice because it is not a brute strength test. And after young mice are exchanged in a control experiment with young blood, 3 out of 4 learn that they have to hang on a tight wire. But if they are exchanged with old blood now, 3 out of 4 do not remember or they cannot do it. And old mice, whether they are exchanged with young blood or old blood, are incapable of improving either agility and coordination or their memory. So they basically just keep dropping down. So to summarize, basically everything before neutral blood exchange, in 2000s, Mike and I had an idea that multiple tissue in old mammals can be systemically rejuvenated and aged. And we published the proof of that in Nature 2005. And the discovery was not about young blood, but the proof of principle that mammalian aging is systemic, right? It's in the blood and it is reversible. So the question to us still remains, how did it happen in blood sharing? What happened? Meanwhile, this is my illustration of an intuitive jump because cats like to jump. Some other groups made this intuitive jump that young blood is the medicine, and they pursued this direction. But we continuously were continuously pursuing the opposite direction, such as that. And it was not really opposite direction, it was simply direction. Why did parabiosis work in the first place? And if you think about it, there really is like a fork in the road. It could have worked because of the young blood, or it could have worked because now we removed, antagonized, or diluted age-elevated negative factors. So when young animal was sharing blood with an old animal, now there is less of those age-elevated proteins. Not only they are physically diluted, but they are removed by the young liver and young kidneys, for example. And then young animal can compensate also and change gene expression of antagonistic proteins. So there are numerous directions that we were investigating. And so then what we discovered is that parabiosis worked because of the old blood dilution. A young blood is not a medicine. And in fact, people who were pursuing young blood as a medicine avenues now run into a sort of the scientific wall because young blood is not working. So now they're switching to exercise-induced molecules, comparing everything in young versus old, are thinking about combination of adding young factors and removing the old, or in other words, to some degree, try to repeat the scientific and IP steps that we have already accomplished. And if you are interested, I can tell you more about it. So this brings me to the most recent discoveries. The main point is that the effect rejuvenation takes place without a putative cause, which is young blood. And even though young blood definitely change with aging, as well as many other things change with aging, this is interestingly not causal for other aging or rejuvenation. So decline of young factors and young proteins happens, but it does not cause aging and it cannot cause rejuvenation. It is a quite interesting scientific conclusion. Because many people ask me continuously to ask by email, It doesn't mean that donating blood would make you young. It is a novel scientific thread for unraveling the cause of age-imposed health decline and a new meaningful medicine. So it's a big scientific direction. It doesn't translate directly that if you drink a lot of water and then use the bathroom very often, or if you donate blood, then you become younger. So this is the paper that we published in May, and then I will end up with the paper that we published in November of 2020. So in the first paper, we show that we can rejuvenate all 3 germ layer tissues just by exchanging all plasma with saline-albumin without any young blood or young blood products. And so this is our small animal blood exchange setup, where we exchange young or old mice with a tube of saline, salty water, with 5% albumin. And there is also blood cells, because once we remove 50% of the old plasma, we then remove albumin, without which we cannot live. And we also remove cells without which we cannot live. So we pretty much then return albumin and cells back, but instead of plasma with the circulating proteins, we now have salty water. And then as a control for procedure, because we use heparin and we use some surgical procedure, we use young exchange and old exchange, where young mice are exchanged with young blood, old mice are exchanged with old blood. As I mentioned, this is miniaturization of the FDA-approved procedure for people, which has very similar, if not identical, principle, and it's called plasmapheresis or therapeutic plasma exchange. What we discovered is that all plasma dilution is safer and more robust for rejuvenation than all other current approaches. That not only young blood is not needed, but then when you perform this procedure, all tissues become statistically the same as young. So they are not statistically different from young. And then on top, now there is some examples. So on top, you see what happens with mouse muscle. So this is mouse procedure. On the bottom, you see what happens with people clinical study. So with mouse muscle, the fibrosis of muscle shown here in this white areas is diminished by one procedure of neutral blood exchange. And we call it neutral blood exchange because our solution does not have age. And then the regeneration, or this dense row of newly formed muscle fibers, which are different color, and they have central blue dots, which is their nuclei, regeneration is improved. So this is a typical for young regeneration, typical for old generation, much worse. And now when young mice are exchanged with neutral blood exchange, the regeneration is dead, similar to young. And old mice exchanged with neutral blood exchange now become similar to young as well, and very different from old. And the same is true for fibrosis. So it is not simply that fibrosis is decreased in old animals after one single procedure of neutral blood exchange, but that now by that parameter, they are not statistically different from the young cohort. And then we can also measure the diameter of the newly formed muscle fibers, and you see how they are not only denser, but they are also bigger, and which is also quantified here. So by many parameters, muscle regeneration is now like young in animals that are similar to 75 to 80-year-old people. And then what we did next, with Dr. Dobry Kiprov, who is our key collaborator on these studies, we obtained blood serum from patients before therapeutic plasma exchange versus after therapeutic plasma exchange. This is post-therapeutic plasma exchange, so it's right after the procedure. And typically blood serum of older individuals is inhibitory for stem cells, which you might need to know because it explains why regeneration declines and why it is through blood. So here you see this one lonely yellow dot as compared to many of these yellow dots on the right. And this yellow dot is one muscle stem cell that is cultured and dividing in old human blood serum. And there are many cells here. So all of the cells that are cultured are shown in blue. But out of all of the cells, only one is attempting to divide here. And then none are dividing in the next in the next part of the figure. So the cells are cultured with old serum, then have decline in their regenerative capacity. Old serum inhibits their ability to divide. But after therapeutic plasma exchange, this inhibition is dramatically less. So now many yellow dots mean that now cells are effectively dividing, even though they are cultured with serum from exactly the same old person, but after therapeutic plasma exchange procedure. And we show that it is unrelated to presence or absence of human serum albumin. So whether we add albumin to cultures or do not add albumin to cultures, there is no effect. So we don't think that albumin is the main factor in our experimental system. And then switching gears, we also demonstrated improved neurogenesis. Now you know what to look for, those red dots at the V region and hippocampus. So now more neurons, many more neurons are formed in the old brains after this procedure. And here is liver adiposity. All of these red dots here are oil droplets or fat in liver. And there is tons of it in control old animals, as well as tons of fibrosis shown here in blue outlines. And then both the adiposity is diminished and fibrosis is diminished by one single procedure. So liver is also healthier. And then now, because many people were studying young blood effects on cognition, not just on the neurogenesis, but cognition, we then more recently published a paper on that as well, where the procedure is exactly the same as I told you. But instead of sacrificing the mice and testing their various tissues, we look at their ability to remember and recognize new texture as compared to old texture and new object as compared to old object. And you probably know that old people, similarly to old animals, are not very curious and inquisitive. So they are not really interested to investigate their environments, and they do not really remember what is new and what is old very well. So by this parameter, by these cognitive parameters, single procedure of neutral blood exchange rejuvenated cognition. So now again, as you can see, old animals are not different from young animals, and they are very different from control old animals that did not have this procedure. And then we always wanted to compare the effects of neutral blood exchange with senolytics, because this is another main profound approach to anti-aging. And so first of all, senolytics work very well. So this is one senolytic, APT263, and senescent cells are shown here in blue by SA beta gal. So there are lots of senescence in old brains. And then senolytic reduces senescence of old brains very dramatically, you can see here. But neutral blood exchange also reduces senescence of old brain, so you can see here. So neutral blood exchange has senolytic or senomorphic properties. So you, in theory, do not need to use senolytics if you use neutral blood exchange. And then we looked at neuroinflammation. This is the last piece of data that I'm going to show you. Neuroinflammation here is illustrated by these red dots in the brain, which label microglia cells. So microglia are not really glia, they're like macrophages in the brain. And when they express this marker, CD68, that means that they are activated and there is neuroinflammation in the old brain. And then neutral blood exchange dramatically reduces neuroinflammation. And that is probably why old mice become rapidly more cognitively capable, because we reduce their neuroinflammation. And then in contrast, if you look at senolytic does not really reduce neuroinflammation, which is quite interesting. So it diminishes load of senescent cells in the brain, but it does not reduce neuroinflammation. It significantly decreases how big these red dots are, but still there are many cells with those red dots which indicate neuroinflammation. So, okay, so then how does it all work mechanistically? And the way that it works is that aging is driven by excess of systemic proteins, which are inhibitory for tissue health and repair. So we cannot evolve out of them because at young levels, they are really indispensable. We cannot live without them. But with age, they become excessive, and it is that excess that then inhibits health and regeneration of multiple organ systems. Consequentially, when we calibrate them, then you become healthier. But what is interesting, and this is the proteomics data, so this is the proteomics data on blood before versus after the procedure. This is mice, and this is people. What happens to the blood proteome after we dilute age-elevated proteins? And what you can see here, that there is lots of red on the right part. There's lots of red here, lots of red here. And if you look at the scale, red means that main proteins become elevated, which is surprising, right? Because we diluted the proteome. How come now we have all of these proteins which are now present at higher level after the procedure? And what you need to realize is that this is 1 week to 1 month after the procedure, is not right after. And the reason that these proteins become elevated is a good reason, is because those are the age diminished, youthful proteome, which we did not really lose, it didn't go anywhere. Those are still encoded in our genome, right, in our genes, but they were physically suppressed by age-elevated circulating factors. So once we diluted them, the repression is now also diminished, and we automatically restore the youthful factors as well. And that produces the second wave of rejuvenation. The first wave comes simply from normalizing the level of age-elevated proteins. But then once their levels are normalized, they cannot repress the youthful proteome, which comes back. And that produces the second punch for improved tissue maintenance and repair. And we show the same specifically for brain proteins. So this is before procedure in mice, after procedure in mice. These groups cluster differently from each other, and the same is true for people before versus after procedure. And these are the identities of the proteins, which then came back and are now elevated. And we have a table in our publication, published in Geoscience in November 2020, on all of these proteins and what do they do for the brain. They all are neuroprotective, and they all improve health of the brain. So I don't know, I can stop here, or I can then tell you why we know that epigenetics also become rejuvenated, and that as well as metabolome, mitochondria, and many other attributes and hallmarks of aging also become younger. But perhaps we could do it in Q&A just to give more time for Q&A. Alison, you just tell me what's your preference.
Well, we do have 4 or 5 questions already. I would just say if anyone in the audience wants to know a bit more about those factors, let us know. And maybe at the end of it, you can.
Yes, I think so too. Yeah, because it's kind of more educational points. It is not really about our discovery.
Okay, lovely.
So the conclusion, right? So this is just, this is the lab, and when we could really socialize and did not have to distance, this is the behind our house, beautiful hills. And this is Mike Conboy, who runs lab with me, and our dog Woofy. This is me. We have excellent lab. We have phase 2 clinical trials underway with Dobry Kiprov as the clinical director, and we do the science. The main conclusion is that large dilution of systemic value stably resets molecular, cellular, and tissue determinants of organs to health youth. And basically doesn't work just on 1 or 2 parameters, but on all of the organ systems. Now I'm on scientific advisory board and have stock option with Juvena, and I hope to launch my company, IMU. So thank you guys for you. Oh yeah. Last but not least, we are very fortunate to be a well-funded laboratory, and these are the funding sources. Thank you guys so much for your attention, and I'll be happy to take questions.
Well, thank you so much for being willing to speak. We have 5 questions already lined up, and I'm sure there's lots more coming. Just tell us when you have to go. I'm just going to start right away with Raphael. Please unmute yourself, and perhaps you can say 1 sentence about where you're coming from, just to give Irina some perspective.
Okay. Hello, Irina. Thank you very much for the presentation. I have to say I've been fascinated by it, by your research since I was aware of it. and I've researched it quite a bit. So I have a question. A first question about it is, what do you think the limits of it are? Because when you present it, it seems quite comprehensive. So what do you expect the limits of the procedure to be?
Well, that's a great question, right? So right now we have clinical studies, and the main point of the studies, there are two main points. One is that how long does one procedure last? So if you have a positive effect, right, then do we need to do another procedure a couple of weeks from then, or can we do it every 3 months or every 6 months? So what is the duration of the positive effects? Since initial positive effects are quite large, then we can reasonably see the decay curve, and we can really, have this conclusion. And the second one, how many age-associated diseases can we tackle? Because official therapeutic plasma exchange, which is FDA approved, is not approved for diseases of aging. It is approved for autoimmune diseases, for example, multiple sclerosis, or rejection of transplanted organs. So for any physician to be able now to advertise and apply this procedure broadly for Alzheimer's or Parkinson's, osteoporosis, and so forth, we need to do the study. So that is the second part, is that what hallmarks of aging can we offset? And by doing this, Raphael, we can then know the limitations. Because right now we tested various things in mice, and we tested effect of human serum on cultured stem cells. So then the limitations will be known after we do the clinical study.
Okay, thank you.
Irina, do you mind if I stop sharing your screen? I know that
if you don't want, so if you, yeah, exactly, I will just exactly.
Perhaps you want to share it again.
Again.
Okay, Jose, you're next.
Thank you for your talk. That was very interesting. I'm a really big fan of your work. So I was interested in the mechanism through which blood exchange is leading to rejuvenation. For example, in this case with senescent cells, is it that there is improved immune clearance of the senescent cells, or is it that the senescent cells themselves are being desenesced, so to speak, back to normal? Thank you.
You guys are asking very insightful questions, by the way. Yes, I believe, or we believe, that they become desenescent or they lose their senescence. We still are investigating what are senescent cells. This is one of the challenges, that they do not have any marker that will not be shared by normal cells that are not senescent. So P16 will be shared by cells which are non-senescent. And even SA beta gal, which is the senescent cell marker, could disappear when cells, for example, are split and are not overcrowded in a tissue culture dish. So what we hope is that by normalizing quickly, acutely systemic proteome, we make many senescent cells less senescent throughout the body. And that's why we see less of them. So it does not work as senolytic. For example, senolytics just inhibit pro-survival molecular pathways in a cell, and therefore senescent cells are dying from that because they're damaged cells. We believe that we simply reverse their senescence in healthier state. It is certainly yet to be determined. That is the hypothesis.
Thank you.
Lovely. All right, next one up, we have Robert.
Hi, yeah, thanks for that very interesting presentation. I just had a quick question of when you mentioned that the old blood makes it worse for the young mice. How much worse? How quickly do they actually get worse with the old blood?
For liver and for brain, they get very quickly worse, like in 1 week, and after one exchange. So, which is quite surprising, right? They have 50% of the young blood still, and they have all of the young organs. So long telomeres, no DNA damage, but simple elevation of age excessive proteome dominantly inhibit neurogenesis, cognition, and liver regeneration in these animals. Doesn't have negative effects on everything. So muscle is still young, but for select organs, the effects are rapid and robust.
So the young animals are resistant to some extent.
In their muscle, they are.
Okay.
Not in their brain. They are very non-resistant. And
Okay,
interestingly, if they are injured, so if muscle is injured, then brain becomes old, much more robustly so, and you know, it's kind of more fragile. And actually, I've heard from neuroscientists, physicians, that they observe the same thing, that if somebody has any sort of peripheral injury and surgery, then there is negative consequences for the brain as well. And it could be because then there is additional inflammation and load on liver and kidney, and then brain basically suffers from that.
Okay, thanks.
Mm-hmm.
Interesting. Thank you. Joshua, you're next.
Irina, thank you for the great presentation. Just a quick question. So how do you think about aging being driven by an excess of systemic proteins versus that excess being sort of a downstream consequence of other drivers, just high level, or as deep as you'd like to go?
Well, of course, excess of systemic proteins is caused by some entropic event in chronology, right? That is for sure. I don't know if that is, I'm looking at my husband there, Mike, who is there, because sometimes he has good points. That, of course, and the excess of systemic proteins initially is caused by damage to cells and tissues. We know that already, right? So when cells and tissues are damaged, then you have certain proteins that are elevated and then they're present systemically. But interestingly, the being driven by systemic proteins means that, let's say that one of your tissue is damaged. Not all of your tissues are damaged simultaneously, but you have a weak link. For example, there is inflammation someplace in your liver. Now, because those proteins are systemic, even though produced by liver, they will propagate aging and senescence to other tissues, making them worse with respect to regeneration and repair. And that is one of the conclusions of our most recent paper, if you guys want to read it, which we published in Neuroscience in 2020. But the general concept, of course, we did not discover it. There is a general concept of systemic propagation of aging and senescence. Does it answer your question? And
Yes,
then you might ask yourself, why do cells become damaged, right? So always every question is a step back to another question, or every answer is a step back to another question. So why do cells become damaged? Unfortunately for us, they become damaged simply because we eat and breathe from oxidative phosphorylation or mitochondrial activity. That is the major cause of damage that we cannot avoid.
And so really small follow-up, what makes these excessive factors then the ideal point to sort of intervene in the biological cascade? Why is this the point to attack, so to say, if you will?
Well, because you cannot stop eating and breathing, right? Can you?
Well, you know, versus no, but I mean versus intervening on, say, I mean, damage removal versus repairing the machinery.
Well, I'm just telling you, Josh, right, that the option to remove all of the damage would be stop eating and breathing, which you cannot do. Since you continue eat and breathe, you will accumulate damage. And the damage cannot be really removed or attenuated at every single level, every enzyme and metabolite, every organelle, every hole in every membrane part of every cell. It is neither feasible nor necessary, because as Plan B, we also developed ability to repair and regenerate. And that is the capacity that is untapped. And what we show is that with a very simple, already FDA-approved procedure, we can make old organs and tissues identical to young or non-distinguishable statistically from young.
Awesome. Thank you so much.
All right, lovely. Next one up, we have Jean.
Hi, Irina. That was actually, that answer was a perfect segue into what I wanted to ask, because my lab is actually working on replacing and removing all the damage that occurs specifically in the brain using cell replacement strategies. So sort of a form of
Cell replacement.
Yeah, sort of a form of regeneration that you mentioned. And so my question is, in looking at, the state of the brain after your treatments, you look at inflammation, increase in or levels of neurogenesis, which are restricted to very small parts, especially the human brain, and also cognition, right? So there are a lot of factors in the literature that have been shown to ameliorate these things and make them look more younger, right? So we even published a paper, genetically manipulating one pathway, showing that we reverse age-associated decline in neurogenesis with just one pathway, and that same pathway reduces inflammation. But when we look at these brains, right, under 2-photon microscopes of the live brains, it's amazing what the difference is between a young brain and an old brain. So you can look in a young brain pretty deeply now with 2-photon microscopy, but by the time they're 4 months, never mind 2 years, right, it's already getting harder and harder. And so despite an increase in neurogenesis, decrease in inflammation, and increase in cognitive function, this opaqueness of the brain as a measure of protein damage and protein aggregates is still going on, unaltered, right? So
Possibly.
have you thought
No,
about that?
Yeah, so good, totally. First of all, thank you so much. These are very, very good points. On the cell replacement and gene therapy, those are their own, interesting technologies with their own challenges, merits, and demerits. So I will wait until gene therapy is effective, and it's much more difficult to people than in mice. And the same goes for cell transplantation. It is very difficult for the brain on numerous levels, or particularly considering that brain connections are who we are. And if we are trying to transplant cells which establish novel connections, that brings about the whole interesting set of problems and questions. Will we be ourselves, even if it works with scalability and health? Yeah. Now, to answer this other part of questions, excellent questions. And we just have recently developed a collaboration with UC Santa Cruz and Stanford to look at various other things under 2-photon microscopy in brain sections. Exactly, what you said. Bunch of neuroplasticity things and synapses and bunch of other things that I'm not going to go into much detail. But to see to what degree did we rejuvenate the brain. Now, to say that after one procedure, old brain will become young, I think it's not realistic. But to say that numerous parameters will be rejuvenated and gradually old brain will become younger is what is our hypothesis postulates. And here I bring this idea of the 2 waves, right? The first wave is simply to reduce SASP, neuroinflammatory proteins, other age-excessive proteins. Then young proteomes come back and does its own thing and gradually improves repair and regeneration. And then if you think about it, we are like 80 kilograms of old tissue that will not become instantaneously younger. But if it becomes gradually younger or does not become older at the same rate throughout, I think that is already a very good first output of the technology.
All right, thank you, Jean. I'm gonna ask Louis' question just because I think he has some noise in the background, but he says, having read many of your great publications, it seems like there are a relatively small number of critical negative factors, maybe a dozen, despite the fact that hundreds of factors may be involved. Do you see additional advantages of depleting negative factors specifically with affinity column versus dilution of all factors?
Yeah, so what we believe, all of these questions from the audience today is just incredibly insightful. And I wonder if you guys are experts in the field and I somehow missed it. And I would certainly look forward if you would like to send me, emails and we can discuss it in more detail. But with respect to hundreds of factors versus few of them, typically there is certain hierarchy or dominance of interactions of key factors or determinants. And they control key nodal points of interactions of signal transduction networks. So really how cell behavior in health and repair is controlled is not by any one factor, but it is not controlled by hundreds or thousands of them. It is controlled by few determinant factors, which then, when present, absent for particular time and particular levels, then determine all of the networks. So if you then identify those key factors and you normalize them to health and youth, they will take care of other hundreds of them. And this is, again, not our discovery. It has been published over many decades on PubMed. In fact, my favorite saying is that if either one of us knew everything that is published on PubMed, we would just discover everything else. But based on our background and expertise, yeah, there are determinants which then control networks of signal transduction pathways with hundreds of the factors. And we don't need to normalize all hundreds.
All right, I'm gonna nod in Louis' stead and say I have a question sneaking in there and also giving other people the space to ask perhaps one or two more other questions. So when I ask you, as I do with everyone, what is your key challenge that you'd like our accelerator projects to solve this year? You just said safely calibrating circulatory milieu to help youth. Could you maybe elaborate a little bit more on that, just in case someone was willing to tackle that?
Yeah, so that pretty much, obviously our current interest in calibrating circulatory milieu, we believe that the way forward is through removal, attenuation, and dilution of age-elevated proteins. But certainly, it is just the very beginning. I mentioned that as a scientific thread to unravel why we age and how to age slower or attenuate the process. So at some point, we have excellent screen systems and excellent computational biology, computation mathematical modeling systems to identify every single determinant age-elevated protein and to see how to calibrate them to precisely young levels, not just in general, but individually, because we do not age identically to each other. So even though there will be the same patterns in our aging, there will be also deviations from those patterns. And so in my opinion, the safe calibration will be precision-based or patient-based, where for every person, we will identify your key proteome, the levels in composition, how does it differ from the healthy young proteome, and how can we reset it now to healthy young proteome. So I think that is a big challenge, which when accomplished will provide meaningful extension of a health span and lifespan.
That's a pretty succinct answer. I think that's a good challenge for people in this group to solve. Okay, we have a few other questions. Let's see how many we can get through. You're very quick, Irina.
That is very, very good. Yeah, it's because the audience is so well informed.
Yeah, I think so. Well, okay, let's see if we can keep it up. Okay, Lynn, you're next. And perhaps a few words about yourself.
Okay, hi, I'm Lynn Cox. I'm based in Oxford, and my lab works on senescence, and we're trying to suppress the SASP because we think that that's probably one of the most damaging parts of senescence. So I was really intrigued by your proteomics data because I was expecting that the cytokines and chemokines would be going down, and yet they're going up. So that was sort of contrary to what I was thinking. And then it made me think, are you actually rejuvenating the immune system? Are you promoting better immunological health? And could that be one of the reasons why you're seeing a loss of the senescent cells? So are you improving senescent cell clearance? And then the final corollary of that is, do you know if your animals have better resistance to infectious diseases? So have you actually improved their overall immunity?
Thank you so much. This is a great question. So first of all, not just animals, but people who right now are undergoing the clinical trial, which is efficacy clinical trial, have improved resistance to infectious diseases, which is great.
Perfect.
It is like really amazing output that we plan to publish the outcomes of the trials in a couple of months, and this is one of them. And we kind of mentioned it anecdotally in the papers that we published, but it seems to hold true. And immune rejuvenation is a main aspect of that, you noticed absolutely correctly. I did not think about it, but you are also completely right that if immune response is now younger, then senescent cells could be cleared. So not only, hypothesis being that they become less senescent and produce less SASP, but some of them, the most decrepit ones, could be also now more successfully eliminated by the immune system.
Okay, no, that's really exciting. I mean, with COVID, we want to be diluting everyone's blood.
I know, but we started actually before COVID. So the first batch of patients started in 2018. And remember that that was before COVID, and many of those samples were on-shelf samples. So they were treated with therapeutic plasma exchange for something else, and we just got access to the repository. But what then we learned is that even though they never had a flu shot, but none of them had flu. So that was like and they're older individuals, and so now this trend seem to hold true.
That's fantastic. Thank you.
So another point, though, it is now some of these people, they did get COVID vaccination, and therefore we have to stop the clinical study because we don't want to dilute the anti-COVID, anti-spike antibodies. So our clinical study could be slightly slowed down by COVID, because when they get COVID vaccinations, obviously, then they cannot be prescribed procedures.
All right, thank you. Well, let's see if we can fit two more in. We have Christine next.
Hi, Irina. It's Christine Peterson, one of the co-founders of Foresight. If I understand the procedure properly, it sounds fairly low risk. And I was wondering, is there any MDs offering this now? In fact, I heard a rumor that the MD you work with in San Francisco does offer it currently.
Yes. So Dobry Kiprov has 25 years of experience with therapeutic plasmapheresis. And in fact, he, I think, pioneered the concept of rejuvenative plasmapheresis and already made a few innovations into the procedure that make it better. So he is our clinical collaborator and perceived chief medical officer of a company that is trying to launch. Now, in general, if you simply use it as prescribed and approved by FDA to treat autoimmune diseases, then there is not many risks. Certainly many fewer risks as compared to injecting yourself with somebody else's body fluids like young plasma. There are some risks though, because your cells are being taken out of your body, spun down in the machine, and then mixed with the saline and albumin and returned back to you. So it is really a complex thing. It is not something that, any doctor could prescribe. So, so I would say that there are risks that are much less than for other alternative procedures, but they could be mitigated if the physicians who are doing the procedure know what they're doing and have been doing it for a while. That would be my, yeah.
Great, thank you.
Well, we have one question here from Alexandra. Maybe you want to ask it too, and I just want to piggyback on there if there's a thing that this group could particularly help with your research. And maybe Alexandra, you want to ask your question too.
Hi, Irina. Thank you again for the update. Awesome new data. I was wondering, if you can tell us anything about future plans, what are you up to in the company, in the university? Anything that you can share with the group?
Yeah, so in the university, we plan to wrap up the scientific results of the current efficacy clinical trial, which is a small trial, let's say 25 people to 40 people, and publish it so people know, the weaknesses and strengths and limitations and the promises of this approach for treating human age-associated diseases. On a company part, basically because I am a university professor, I was looking for a business person to spearhead the company. And finally, I was reached by email, and now we have excellent business person who started a few multi-million-dollar companies in the recent past. And so he is basically connecting us with potential investors, and we give the introductory talks and slide decks presentations. The idea for the company would be to launch the company at the time when the results of current clinical trials come out, and to have funding for larger phase 3 clinical trials simultaneously as the first step of the company launch. And in the phase 3 clinical trial, we need to have placebo group randomization. And right now, I have to mention it. Thank you so much, Alexandra, for bringing it up. Right now, our clinical trials are longitudinal. So we do not have placebo group. We just compare before and then after one round of procedure, 2, 3, 4, and so forth. And then how long it does it take for the positive effects to decay. So we have longitudinal clinical trial comparison, which is IRB approved on a national level, but the participants are paying for procedures and are paying lots of money. So that's how current clinical trial is funded. And that's how it was approved by IRB. So then at the launch, which we hope to have in May, we would need to have investment large enough to support phase 3 clinical trial where participants do not pay. And then we have more people, and then we have placebo group control. And we still plan to have the service for fee branch of the company to continue with something that we have proven to work already. So that's like in a nutshell, you know, yeah, what we hope to accomplish. Yeah.
Well, very specifically, what could people in this group do that would majorly help?
Well, networking and advice, particularly in commercial realm, would help a lot. So then, you know, instead of making our own mistakes, you guys can tell us where to look and not to make them, how to troubleshoot. And then, of course, you know, the major help will be with the resources, with the resources for phase 3 clinical trial. Because that is the very first, I think that is the priority. We have longitudinal trial success when people pay it. We absolutely have to be, you know, to have good standard of research and medicine to say that now we have phase 3 clinical trial, which after successful completion, we can approve this procedure by FDA for new classes of diseases, advertise it broadly, and perhaps have health insurance involved and compensated some of the procedures. So yeah, so resources and networking, that would be a huge help.
All right.
Irina, what's the high level? It's great that you're trying to commercialize this, and it's great that since it's already a procedure in humans that you can jump right to late stage trials. But what's the high level story on why you can commercialize it? What can you protect in terms of intellectual property? Like, since it's already approved, how can your company gather the money for it if you fund the trial?
Great question. So it is approved for completely different class of diseases. So if a clinician just wants now to start making money of it, or anybody, prescribing it for Alzheimer's, Parkinson's, osteoporosis, or anti-aging, then they will have problems with FDA. They will have FDA warnings. And the only way to go forward, in fact, is to perform clinical trial to show that the procedure is effective for those classes of diseases. Now, in doing our clinical trial, we already know that the procedure has to be modified. We call that therapeutic plasma exchange plus. So it is not the identical procedure to the one that has been approved, but it is repositioning of other FDA-approved processes and medicines. So it is not really novel chemical composition, and that is the first step in our intellectual property protection, is trade secret.
So you think you can protect the combination of tweaks?
Combination of tweaks is really trade secret protection. However, there is a very tempting possibility to repatent for new application of patent previously FDA approved and off patent procedures, medicine, device, so forth. So that is the easiest thing to do. And then of course, there are numerous avenues for changing the composition of exchange fluids, such as that chemical composition itself is IP protected, to make the procedure last longer, to encompass more hallmarks of aging reversal. So that is the third way. So we really start by, notoriety. We are inventors and we already have numerous tweaks, which we know how to apply successfully. And then the second wave is repatenting of patent things. And then the third coincidental wave is now novel chemical composition of exchange fluid. And so that is within like the first couple of years. And then, of course, sky is the limit, because we just discussed here that we can identify for each person the precise combination and concentration of things to be removed and to be restored. And there are additional patents on how to do it safely. So that's like 7-10 years so far. Did I miss anything? Cool.
Well, we do have 3 more questions. I'm just going to read them out to you, and you just decide if you want to take any of them or none of them. Okay. All right. So we have, what do you think about existing clinics in Europe for plasmapheresis? Then we have, do you have any idea of the molecular weight cutoff of the molecules you think are needed to be removed? And how long do the benefits last? And so how often do you require treatment?
Right. So the same thing about existing clinics. Some people who are in Europe, they are in fact get in touch with us, and they tell us that, well, we potentially can do this, but we don't really know exactly how to do it. We have no idea how to apply it to anti-aging at all, because it is not completely benign procedure. Would you guys affiliate with us? So you teach us, provide service and support, and we give you share of profits. So Dobry Kiprov now has developed excellent affiliation plan, and there are at least 4 clinics in US and a couple in abroad who are interested in affiliation. So they pay upfront fee, and then we pretty much train them and provide support, and the patients are actually our patients, so we are working on that. Then with respect to molecular weight, no, there will be proteins of all kinds of molecular weight, and they will be age elevated, so you cannot simply have a pour of 20 kilodalton and remove aged factors. And that's why it is really was laughable and not serious to find a working fraction that will make you younger. There is no silver bullet, one fraction. There are a combination of proteins, and we published on those as well. So sorry if I forgot. What other questions?
Thank you. No, you remember, you answered them very well. Hey, thank you so much. I think we can all just thank you.
Thank
very
you guys.
much for
Yeah, please stay in touch.
Long.
Much appreciated your interest and even getting in touch with us. Bye-bye.
Yeah, please let us know how we can help. Thank you so much. I'm going to post a link to those of us who want to stay on longer and socialize in a different room. No worries about you, Irina. Please feel free to drop off. Please let me know anything that I can follow up with the group with, and vice versa. Thank you very much.
Have a good day.
Bye. All right, everyone. For those of you who still want to continue chatting, then I just posted a link in the chat. And I see many of you in the gather room. It's best to join via Chrome. You can also now join via Safari, but this is a space for us to just all kind of debrief on this meeting. And yeah, thank you all for joining. This was fantastic. And I'll see you next month for the next proper one. But I'm hoping to see many of you in the Hangout afterwards. See you in a second, and bye-bye.